HCI has witnessed an increasing body of work geared towards engaging modern human living organisms in design, alongside renewed approaches to more conventional fields of bioart and biodesign. At the same time, we have seen the expansion of critical approaches towards decentering human perspectives and giving ethical consideration to modern human living organisms and species in design. In this paper, we bridge the two areas of research with a focus on revealing issues and attempting to foster reflection on practical ethical considerations in working with microorganisms in both laboratory and studio practices. In particular, drawing from the critical literature on modern human labor and non-participation, we propose a provocative notion of microbial revolt, which aims to redefine tools, spaces, and practices in biology labs and studios to review embodied forms of exploitations, to create space for non-participation, and to foster modern human care ecologies. This exploration took the form of a workshop involving both biologists and designers working with non-human species in their daily practice, followed by an interview stage in which some of the ideas that emerged from the previous discussion were discussed more thoroughly with individual practitioners. The Microbial Revolt workshop consists of two parts. First, participants were guided to write a comparative modern human journal from the perspectives of lab organisms in lab and feral environments. Next, drawing from the inspiration of design movement against utility and consumerism in the 90s, Jintoku, the participants are asked to redesign lab equipment into futile objects as a form of critical making to facilitate revolt of the role-played organisms, with a set of prompts of lab equipment and forms of revolt. Examples of the tension surfaced by the journaling exercise include controversies between feral or lab environment preferences. While designers tend to see lab as less ideal and decontextualized, biologists portray it as a more caring environment. When it comes to the Jintoku, we observed analogous idiosyncrasies. Despite directions, only one biologist redesigned the lab equipment to support the microbial revolt. In all other cases, modification would take place in the organisms, such as changing their chromosomes to break experiment reproducibility. In contrast, designers tend to actively challenge the original purpose of the equipment by changing its form, structure, scale, or material. Another mode of revolt explored by designers looked into the suppressed characteristics or ecological interactions of the organisms in a lab setting. Based on the data collected during the Microbial Revolt workshop, we synthesized a number of initial insights. First, biologists' and designers' responses reflect different epistemic cultures. While conventional lab environments prioritize control and reproducibility, design practices appeared more open to considering wicked problems characterized by a high degree of complexity and contextuality. Furthermore, biologists tend to view microbial revolt as anarchist threat addressed to the scientist, while some designers interpret it as an expression of unmet needs, which can still be accommodated through mutually beneficial arrangements. These initial insights led to the question, which we explored more deeply with a round of semi-structured interview. How are these epistemic cultures negotiated by practitioners engaging with spaces, tools, and guidance that originate in biology research? What are the perceived power dynamics? How can ecological perspectives be expanded? As we interviewed practitioners in biodesign and bioart, we aimed to further investigate how one might navigate these issues and balance secluded and ecological research approaches in future bio-related practices. We grouped the interview results in four main themes. First, most designers saw traditional biolab environments and practices as narrowing the scope of work and even creative capacities, Often, the suppression of creativity was linked to the lab environment itself, the equipment at hand, or the lab regulations. Second, while some practitioners did think about the biolab environment as facilitating care and attention, they also acknowledged the limitation of the nature of care in lab settings. Other participants reflected on the productionist nature of care practice in the lab. Some also warned against the potential risks of uninformed care. Next, in terms of ecological thinking, uncritically adopting life science knowledge into the design field are seen by some participants as promoting organism decontextualization and control. Some try to counteract such effects through recontextualizing the organisms. Some also try to promote a more naturalistic approach, such as by following the organisms in their work. Regarding new tools and protocols, participants provided examples of tactical approaches to develop their practice within current regulatory frameworks, such as the creation of a liminal zone, or by proposing alternative tools to accommodate working with environmental samples in more naturalistic ways. Some also proposed a more cooperative negotiation with safety or ethics regulators. Based on the interview and workshop results, we concluded 3 main topics for discussion. First, we discussed practices of resistance and microbial revolt. Although speculative, the angle of revolt serves as a critical tool to reflect on the power dynamics between humans and modern humans in lab and studio practices. Thinking about non-participation and revolt helped to redirect participants' attention to the characters of the organisms, ultimately exposing and/or reimagining utilitarian tools and modes of working. Second, we discussed epistemic remoteness versus intimacy and care ecologies in the lab. Lab environments, while providing controlled conditions that eliminate noise, can hinder contextualizing the experiments within wider ecological and social scenarios. The highly mediated interaction between organisms and researchers promotes a culture of objectivity and rationality that creates distance and restricts alternative engagement modes or deeper ethical relations. Designers that took part in the interview shared efforts to counteract remoteness and support with more nuanced care practices of attentiveness, responsibility, and responsiveness. Finally, we discussed laboratory protocols versus creative freedom. In the interviews, concerns were raised about tensions between laboratory protocols, creative freedom, and modern human care ecologies. While conventional biolabs guarantee greater safety and resources, they also come with stricter rules and potentially more conflicts with creative practices. DIY labs and more studio-based space can provide a higher degree of freedom while carrying more unknown risks brought by new tools, practices, and a diversity of species. Therefore, we identify a need for a dedicated management framework for tools and practices in creative settings. Following the discussion, we draw 5 practical implications for design and HCI research. First, reframing sterilization. There is a clear demand for an increase in the flexibility of sterilization mechanisms for appropriation in design contexts, such as by adopting naturalistic probiotic approaches to create selective environments in collaboration with microbial communities. Second, increasing and systematizing documentation of design-based practice. We propose better documentation of novel lab practices in design as a strategy that informs practitioners and provide more tangible materials for discussion, which might assuage regulators' concerns over safety risks or bioethical issues. Third, defining new frameworks for risk management. We draw attention to the need for new guidance that accommodates ecological perspectives, phenomenological practices and sensibility, and more varied ways of engaging with organisms, specifically in design scenarios. Fourth, Designing liminal spaces for heterogeneous areas of research. We identify opportunities in the concept of liminality, the creation of new contact zones between spaces with different protocols, through the assemblage of new spaces that fuse the benefits of design studios and biolabs, while circumventing their limitations. Finally, we propose Revolt as a way to expand perspectives in modern human HCI. Within the wider HCI context, the notion of revolt and modern human resistance could be introduced as a provocation to extend case studies of interspecies design, serving as a tactical approach to more radically engage modern human interest against anthropocentric dominance. In conclusion, in the growing trend of considering community involvement and mutuality in design, our method based on revolt, non-participation, and resistance can represent a powerful tool to scrutinize the power dynamics underlying the involvement of modern human organisms and to establish fairer grounds for co-creation with them. It is only by accepting and making space for potential resistance that we can acknowledge different perspectives and the labor of all parts, and be able to create space for bidirectional negotiation, adjustment, and care among humans and modern human participants involved in our designs.